हेलो डियर स्टूडेंट्स, वेलकम टू बायो क्लासेस ऑनलाइन। आज मैं लेके आया हूँ मल्टीपल चॉइस क्वेश्चंस, दैट इज MCQs with the explanation of all the four choices from the chapter Cell, the Unit of Life. These MCQs are very important for NET and NEET 2020. Let's start. First of all, we will take the MCQs from cell theory and overview of cell. Let us have a look on the first MCQ. Unicellular microscopic organisms were first studied by Robert Hooke, Priestley, Pasteur, and Leeuwenhoek. Robert Hooke introduced the term cell in his book Micrographia in 1665. Later on, it was found that the structures Robert Hooke found were the cell walls. So Robert Hooke is not the correct answer. Priestley discovered oxygen. So this answer is not also the correct. Louis Pasteur discovered principles of vaccination, microbial fermentation, and pasteurization. So again, this Pasteur is not the correct answer. So the fourth option is definitely the answer, that is Leeuwenhoek. He was the scientist who discovered the single-celled organisms, whom he called the animalcules, he is considered as the father of microbiology. So the right answer of this question is Leeuwenhoek. Next question. The microscope usually used for seeing living cells or tissues is compound microscope, electron microscope, phase contrast microscope, light microscope. Compound microscope and light microscope are one of the same thing. They have low resolving power, and the specimen which we want to observe under the compound microscope or light microscope or bright field microscope are to be killed before seeing under these microscopes. So these compound microscope and light microscope is not the correct answer. Electron microscope uses the electron beams to form the image of an object or a specimen and have a high resolving power. But here again, the specimens, the structures or the subcellular structures which we want to see under the electron microscopes are to be killed, because the electron microscope, in electron microscope there is vacuum, there is no air. So the answer is this phase contrast microscope. Phase contrast microscope is an optical microscope that converts the phase shifts in light passing through a transparent specimen to brightness changes in the image. So the answer is phase contrast microscope. Next question. The most likely method used to determine the structural details of the cell organelle is? Which one of the method is used to determine the structural details of the cell organelle? Options are autoradiography, microdissection, electron microscope, and phase contrast microscope. Autoradiography is a technique using X-ray film to visualize molecules or fragments of molecules that have been radioactively labeled. ऑटोरेडियोग्राफी एक टेक्निक है जिसमें हम एक्सरे फिल्म यूज़ करते हैं टू विजुअलाइज द मॉलिक्यूल्स और फ्रैगमेंट्स ऑफ द मॉलिक्यूल्स जिनको ऑलरेडी रेडियो एक्टिव लेबल्ड है. सो दिस इज ऑटोरेडियोग्राफी इज नॉट द करेक्ट आंसर. Microdissection refers variety of techniques where a microscope is used to assist dissection. माइक्रो डिसेक्शन वैरायटी ऑफ टेक्निक्स होती है जहाँ पे हम माइक्रोस्कोप को यूज़ करते हैं टू असिस्ट द डाइसेक्शन. इलेक्ट्रॉन माइक्रोस्कोप uses the electron beams and has a very high resolving power and can be used to determine the structural details of the cell organelle. So the correct answer is electron microscope. Phase, as we know in the previous question, phase contrast microscope is an optical microscope, is used to visualize, to study the living cells, but not the cell organelles, because this phase contrast microscope does not possess a high resolving power. So the correct answer to this question is electron microscope. Next question. The figures of cork cells as seen by Robert Hooke were published in a book. जो रॉबर्ट हुक ने फिगर्स कॉर्क सेल्स जो ऑब्जर्व करा था अंडर द माइक्रोस्कोप, उसने उन फिगर्स को किस बुक में पब्लिश किया? The four options are origin of species, species planetarium, genera planetarium, and micrographia. Origin of species was published. The author of the origin of species is Charles Darwin. So it is not the correct answer. The author of species planetarium is Carl Linnaeus. So again, this is not the correct answer. The author of genera planetarium is again the Carl Linnaeus. And the fourth option, micrographia, is written by Robert Hooke. And this is the correct answer, that is the micrographia. So we can say the figures of cork cells as seen by the Robert Hooke were published in the book Micrographia. Next question. Who proposed a modification in a cell theory? Options are Schleiden and Schwann, Rudolf Virchow, Robert Hooke, Marcelo Malpighi. Schleiden and Schwann has given the cell theory. They have not given the modification in the cell theory. Schleiden and Schwann states that the new cells, they were not able to state that where from the new cells arise. So they are concerned with the cell theory. Rudolf Virchow has given the modification in the cell theory and has given a statement omnis cellula e cellula, that is new cells arise from the pre-existing cells. So this is this is the correct answer, Rudolf Virchow. Robert Hooke, as we know, is concerned with the introduction discovery of the cell and has published the book Micrographia. Marcelo Malpighi discovered the invisible world of human body and plants by studying tissues under microscope. He is considered as the father of histology, and this Malpighi discovered the cells prior to Hooke, who called them sacules and utriculus. So the next question is omnis cellula e cellula. This statement was given by, as I already mentioned it in the previous question, this statement, that is the new cells arise from the pre-existing cells, was given by the Rudolf Virchow. So answer is Rudolf Virchow. In this statement was given in 1859. Schleiden and Schwann are has given the cell theory. Robert Brown discovered the nucleus from orchid root, and Robert Hooke coined the term, or rather introduced the term, cell. Next, dash are the exception to the cell theory. Bacteria are made up of the cells, or are cellular in nature, fungi are cellular in nature, lichens are the compound plants as algae and fungi live together in close association, as a result of which both are benefited. This relationship is called the symbiosis. Lichens एक symbiotic association है algae और fungi की, so they are also cellular in nature. It is only the viruses which are acellular in nature. When they are acellular in nature, so this is an exception to the cell theory. So the correct answer is viruses. Next question. Schleiden and Schwann are connected with. We know that Schleiden and Schwann are connected with the cell theory. Theory of cell lineage, that is omnis cellula e cellula, which was given by Rudolf Virchow. Cell theory was given by this Schleiden and Schwann. Protoplasm as the basis of life, this was given by the Huxley. So the correct answer is cell theory. Next question. Cellular totipotency is related to synthesis of new cells, formation of new cells, capacity of a cell to form a whole organism, formation of new species. Cellular totipotency, in the previous lecture I have mentioned it, that it is the capacity of a cell to form the whole organism. So the correct answer is C. Synthesis of new cells or formation of new cells occurs by means of the cell division, and the formation of new species is called as speciation. Question next. Who first of all proposed a cell to be a unit of life and component of all tissues? Schwann, Schleiden, Deuterochette, Lamarck. So किसने प्रपोज किया था कि जो सेल है दैट इज द यूनिट ऑफ लाइफ एंड कंपोनेंट ऑफ ऑल टिश्यूज? Schwann studied that all animal cells are made up of the cells and the components and its components, while the Schleiden was a German botanist concluded that all plant tissues are made up of cells. Deuterochette discovered osmosis and also stated that all organic tissues are really globular cells of an extreme smallness which are united by cohesion. So Lamarck is concerned with the evolution and has given the theory of organic evolution, that is use and disuse of organs. But here the Schwann proposed the cell hypothesis also, and it is the Schleiden who first of all in 1838 Schleiden concluded that the cell first, before Schwann, it was the Schleiden who concluded that the cell to be a unit of life and components of all tissue. So the answer is Schleiden. Next question. A plant cell has potential to develop into a full plant. This property of plant cell is called as tissue culture, totipotency, pluripotency, gene cloning. As we know, the capacity of a plant cell to form a whole full-fledged plant is called as the totipotency. The answer is totipotency, but here the three options: tissue culture means the growth of tissues or cells in an artificial medium separate from the organism. टिश्यू कल्चर का मतलब हुआ कि टिश्यूज और सेल्स को ग्रोथ करना आउटसाइड द ऑर्गेनिज्म इन एन आर्टिफिशियल मीडियम. प्लूरिपोटेंसी का मतलब हुआ कैपेसिटी ऑफ स्टेम सेल टू गिव राइज टू द सेवरल डिफरेंट सेल टाइप्स। प्लूरिपोटेंसी का मतलब हुआ कि स्टेम सेल जो होती है जो वो डिफरेंट टाइप्स ऑफ सेल्स दे सके, उस कैपेसिटी को हम कहते हैं प्लूरिपोटेंट। सच एज वी कैन से द हेमेटोपोइटिक stem cell is a pluripotent cell, that is the blood stem cell is a pluripotent cell that can give rise to different kinds of the cells such as RBCs, WBCs, neutrophils, basophils, eosinophils, etc. And the gene cloning is the recombinant DNA technology where we insert a gene of interest in a particular vector. So the answer is totipotency. Now we will take some important questions from the prokaryotic cell. Slime layer and capsule in bacteria. Slime layer and capsule in bacteria are modifications of number 1, Glycocalyx, cell wall, plasma membrane, pellicle. Here the answer is Glycocalyx. It is the outermost layer made up of macromolecules. It protects the cells and also helps us in adhesion. If this layer is in the form of a loose sheet, it is called the slime layer. And if this layer is thick and tough, then it is called the capsule. But the cell wall, we know that this is the this is one of the covering in case of the bacteria. It is made up of the peptidoglycan, and plasma membrane is made up of the lipids and proteins. So the answer is Glycocalyx. Next question is the enzyme peptidyl transferase of prokaryotes resides in the कौन सा एंजाइम होता है जो ये एंजाइम जो पेप्टाइडाइल ट्रांसफरेज होता है वो किस पे होता है? As we know, the ribosome of prokaryotic cells is 70S type, and the subunits of the 70S type ribosome are 50S and 30S type. So this is 40S and 60S ribosomes are the subunits of eukaryotic ribosomes. So the C and D is not the answer. So we have to find out the answer from the 50S and 30S ribosome. So it is the 50S ribosome which is possessing this enzyme peptidyl transferase, the enzyme which transfers the amino acids to join with the rest of the chain of the amino acids. So the answer is 50S ribosomes. Next question, which of the following is always absent in prokaryotic cells? प्रोकैरियोटिक सेल्स में इनमें से कौन सा हमेशा एब्सेंट होता है? प्रोकैरियोटिक सेल्स में राइबोसोम्स होते हैं 70S टाइप, सो दिस इज नॉट द आंसर. Mitochondria is a double membrane bound organelle. Prokaryotic cells do not possess the membrane bound organelle, so the answer is here mitochondria. In prokaryotic cells DNA present होता है in the form of a nucleoid, and cell wall is also present in prokaryotic cells, that is made up of the peptidoglycan. So the answer here is the mitochondria. Next question, the type of ribosome found in the prokaryotes is? राइबोसोम्स जो प्रोकैरियोटिक्स में होते हैं वो 70S टाइप होते हैं and are made up of 50S type and 30S type subunits. So the answer is 70S type, while as 80S type is present in eukaryotic cells. 30S is one of the subunit of the 70S type ribosome, and 50S is another larger subunit of the 70S type ribosome. So the answer is 70S type ribosome. Next question, if you remove the fimbriae from the bacterial cell, which of the following would you expect to happen? First of all, we will come to know what are the fimbriae. And these fimbriae are these fimbriae are small bristle like fibers coming out of the cell. ये bristle like structures fibers होते हैं, and there are mainly there are 300 to 400 fimbriae per cell, and they help the bacteria to attach to solid surfaces such as rocks and host tissues. So अगर ये इसका इन फिम्बरी का काम होता है, दे आर ब्रिसल लाइक फाइबर्स होते हैं, 300 से 400 बैक्टीरिया सेल में पर बैक्टीरिया सेल में होते हैं, और हेल्प करते हैं, ये हेल्प करते हैं बैक्टीरिया को टू अटैच द सॉलिड सरफेसेस, रॉक्स हो या होस्ट टिश्यूज हो. अगर ये रिमूव हो जाते हैं बैक्टीरिया सेल से, सो द बैक्टीरिया विल नॉट बी एबल टू अटैच टू द होस्ट टिश्यू or the rock surface. So let's find here in the options. The bacteria could no longer swim. Swimming के लिए जो बैक्टीरिया की लोकोमोशन के लिए होता है, that उसके लिए वो flagella होता है. Bacteria would not adhere to the host tissue. So this is the right answer, second one, because these are responsible for, these fimbriae are responsible for attaching the bacteria to the rock surfaces or host tissue. ट्रांसपोर्टेशन ऑफ मॉलिक्यूल्स अक्रॉस द मेम्ब्रेन वुड स्टॉप. दो ट्रांसपोर्टेशन के लिए चैनल्स होते हैं, कैरियर प्रोटीन्स होते हैं, तो वहाँ पे कोई जरूरत, द शेप ऑफ बैक्टीरिया वुड चेंज, देयर विल नो चेंज इन द शेप ऑफ द बैक्टीरिया if the fimbriae are removed from the bacterial cell. So the answer is the bacteria would not adhere to the host tissue. Next one, which of the following is seen only in प्रोकैरियोटिक सेल? इनमें से कौन सा ऐसा स्ट्रक्चर होता है जो सिर्फ बैक्टीरिया सेल्स में होता है? डिक्टियोसोम्स आर द गोल्जी बॉडीज प्रेजेंट इन द प्लांट्स. सो दिस इज नॉट द करेक्ट. राइबोसोम्स आर प्रेजेंट इन बोथ प्रोकैरियोट्स एज वेल एज यूकैरियोट्स. एज वी नो प्रोकैरियोट्स में 70s टाइप होता है, यूकैरियोट्स में 80s टाइप होता है. एंड mesosomes are the structures or the infoldings of the plasma membrane of prokaryotic cells and are responsible for the respiration in them, and they are considered as the mitochondria of the prokaryotic cell. And this is the correct answer, mesosomes. It is only found in the prokaryotic cell, not in the eukaryotic cell. Endoplasmic reticulum is again a membrane bound cell organelle present only in the eukaryotic cells. So next question, in eubacteria, a cellular component that resembles eukaryotic cell is, this question has been asked in AIPMT 2011. कौन सा ऐसा सेल्यूलर कंपोनेंट यूबैक्टीरिया, दैट इज द ट्रू बैक्टीरिया, जो यूकैरियोटिक सेल्स से रिजेंबल करता है? देखिए, सेल वॉल जो बैक्टीरिया में होता है, दैट इज मेड अप ऑफ द पेप्टाइडोग्लाइकन, वाइल एज अगर हम यूकैरियोटिक सेल सच एज प्लांट सेल की बात करें या फंजे की, प्लांट्स में तो सेल्यूलोज से बना होता है और फंजे में काइटिन से बना होता है. तो यहाँ पे तो ये फर्स्ट इज नॉट द राइट आंसर. प्लाज्मा मेम्ब्रेन जो होता है, that is similar, that is according to the fluid mosaic model, the prokaryotic and eukaryotic plasma membrane are similar in composition. So the right answer is the plasma membrane. Nucleus is only present in the eukaryotic cells, and the genetic material in prokaryotic cells is in the form of a naked structure called the nucleoid, and the ribosomes in case of prokaryotes are 70s type, while as in eukaryotic they are 80s type. So the answer is plasma membrane. Next question, the mobile bacteria are able to move by, this question has been asked in All India CBSE 2014. मोबाइल जो बैक्टीरिया होता है, मूवमेंट ऑफ बैक्टीरिया इज कैरिड आउट बाय सीलिया, पिल्ली, फिम्ब्री, फ्लैजिला. सीलिया नहीं होता है, पिल्ली आर रेस्पॉन्सिबल, सीलिया आर mainly absent in the prokaryotic cells. These pili are elongated tubular structures made up of a special protein called pillin. there are mainly 1 to 4 pili per cell, and they help in the conjugation, transfer of the genes from the plasmid to from donor to the recipient, जिस प्रोसेस को हम कहते हैं conjugation, or these pili are responsible for formation of a conjugation tube between the two bacteria, and through this conjugation tube the genetic material mainly from the plasmid is transferred from the donor to the recipient. And so this is not the correct answer. Fimbriae, as I already mentioned, that they are responsible for the attachment of the bacteria to the solid surfaces such as rocks, such as host tissues. So this is not on, it is the flagella which helps the bacteria to move, to become mobile. So flagella is the right answer. Next question. Which one of the following living organisms completely lacks a cell wall? इनमें से कौन सा बैक्टीरिया, कौन सा ऑर्गेनिज्म है दैट लैक्स ए सेल वॉल? एंड हियर द ऑप्शंस आर सैकेरोमाइसिस, ब्लू ग्रीन एल्गी, साइनोबैक्टीरिया, सी फैन, gorgonia. And Saccharomyces are the fungi and possesses the cell wall made up of the chitin. So it is not the right answer. Blue green alga and cyanobacteria are one of the same. They are the bacteria and possess the cell wall. So they are not the sea fan belongs to the kingdom Animalia, and the animals possesses the do not possess the cell wall. So this is the right answer. Next question. Which structure perform the function of mitochondria in bacteria? And this question has been asked in All India CBSE 2014. Cell wall कौन सा स्ट्रक्चर बैक्टीरिया में होता है जो एक्ट करता है जस्ट लाइक एज द mitochondria in bacteria? Cell wall is not, cell wall helps the bacteria from getting desiccated. This is not the mesosomes are the enfoldings of the plasma membrane and are responsible for the respiration and are considered as the mitochondria of bacteria. So this is the right answer. Nucleoid is the genetic material clumped in the center of the bacterial cell. It is the naked without covering, without any membrane covering. So it is not the right answer. Ribosomes are responsible for the formation synthesis of the protein. So ribosome is not the right answer. Next question. Which of the following structure is not found in the prokaryotic cell? Again, this question has been asked in the All India CBSE 2015. The options are ribosomes, mesosomes, plasma membrane, nuclear membrane. कौन से स्ट्रक्चर्स होते हैं जो प्रोकैरियोटिक सेल में नहीं पाए जाते हैं? Ribosomes are present in the prokaryotic cell. So this is, as we know, 70s type होता है. So this is not the right answer. Mesosomes होते हैं, they are the enfoldings of the plasma membrane of the bacterial cell. This is not the right answer. Plasma membrane is also present in the prokaryotic cell. Again, it is the nuclear membrane. It is the nuclear membrane which is not present in the prokaryotic cell. So this is the right answer. Next question. The structure that helps some bacteria to attach to the rock and or host tissue are. This has been asked in All India CBSE 2015. इस क्वेश्चन का तो आपको आंसर डेफिनेटली पहले ही पता होना चाहिए क्योंकि मैंने मेंशन किया है. these are the स्मॉल ब्रिसल लाइक फाइबर्स जिनको हम फिम्ब्री कहते हैं. 300 से 400 फिम्ब्री होते हैं पर सेल और यही हेल्प करते हैं बैक्टीरिया को टू अटैच द सॉलिड सरफेसेस सच एज रॉक्स एंड होस्ट टिश्यू. सो फिम्ब्री इज द राइट आंसर. So, but mesosomes, as we know, they are the enfoldings of the plasma membrane of the prokaryote. Hold fast. What is hold fast? Hold fast is a root-like structures by which an aquatic sessile algae are attached to the substrate. Hold fast root-like structures होते हैं जिससे non-mobile, non-motile aquatic algae अटैच होते हैं सबस्ट्रेट के साथ. सो दिस इज नॉट द राइट आंसर. Mesosome is not the rhizoids. Rhizoids क्या होते हैं? Rhizoids are structures in plants and fungi that function like roots in support or absorb. ये rhizoids होते हैं root like structures और किनमें प्लांट्स और फंजे में और हेल्प करते हैं सपोर्ट और फंक्शन में. सो राइजोइड इज नॉट द राइट आंसर. नेक्स्ट क्वेश्चन. विच ऑफ द फॉलोइंग इज नॉट ए फीचर ऑफ ए प्लास्मिड? व्हाट आर प्लास्मिड्स? Plasmids are the extra chromosomal genetic materials in the bacterial cell. Extra genetic materials in the bacterial cell or the prokaryotic cell. They are extra chromosomal. They are the extra nuclear. These plasmids are the extra nuclear DNA. They possesses the genes for fertility. So they are where they are called the F factor. They possess the genes for resistance towards the antibiotics. They are called the R factors. They are single circular double helical genetic material. So which of the following is not a feature of the plasmid? This question has been asked in NEET 2016. Single stranded these plasmids are double helical structures, double helical circular structures. So they are not single stranded. Independent replication, they are single. They are not. Which one of the following is not a feature of? इनमें से कौन सा है जो फीचर नहीं है प्लास्मिड में? They are single stranded. No, they are single but double helical structure. They are not single, they are double helical structure. So this is the right answer. Independent replication, they show the independent. They are not dependent on the replication of the nuclear genetic material. So they show the independent replication. This is the correct about the plasmid. So this is not the answer. Circular, they are also circular. This is the correct. This is not the feature of this is the feature of this plasmid. So this is not the right answer. Transferred, they are able to get transferred from the donor to the recipient through a conjugation tube. So this is not the right answer. So the answer is the A. They are not single stranded. Next question. One of the major component of the cell wall of most fungi. It has been also again asked in NEET 2016. Hemicellulose, chitin, peptidoglycan, cellulose. Hemicellulose is the कौन सा ऐसा कंपोनेंट है जो सेल वाला फंजे में नहीं सेल वाला फंजे में होता है? हेमिसेल्युलोस प्लांट सेल्स में होता है. दिस इज नॉट द राइट आंसर. पेप्टाइडोग्लाइकान इज present in the bacterial cell wall. This is not the right answer. Cellulose is present in the plant cells walls. Again, this is not the chitin. Chitin is a polysaccharide made up of N-acetyl glucose amine. So chitin is the right answer. Next question. Antibiotic resistance gene in bacteria are located on. पहले ही जब मैंने बताया था कि जब कंजुगेशन होता है वहाँ जो प्लास्मिड्स होते हैं, प्लास्मिड्स में कुछ प्लास्मिड्स होते हैं जिन प्लास्मिड्स में एंटीबायोटिक रेजिस्टेंस जीन्स होते हैं उनको हम कहते हैं एंड दे शो द रेजिस्टेंस टुवर्ड्स द एंटीबायोटिक्स. उन प्लास्मिड्स को हम कहते हैं आर फैक्टर और आर प्लास्मिड्स. सो यहाँ पे बताया जाता है कि कौन सा कहाँ पे एंटीबायोटिक रेजिस्टेंस जीन्स होते हैं. दे आर प्रेजेंट ऑन द प्लास्मिड्स. प्लास्टिड्स तो नहीं, बैक्टीरिया में होते ही नहीं है, न्यूक्लियाइड जीन्स में नहीं होते हैं और मीजोसोम्स पे नहीं होते हैं. और यहाँ पे एक लास्ट पॉइंट आपको मैं यहाँ इस प्लास्मिड के बारे में, देर आर सर्टन प्लास्मिड्स हुज फंक्शन इज आर स्टिल अननोन. जिनके अभी भी फंक्शन अननोन है उन प्लास्मिड्स को हम कहते हैं cryptic plasmids. So here the answer is plasmids. Next, nucleoid or genophore consists of, जो न्यूक्लियाइड या जिसको हम जीनोफोर कहते हैं बैक्टीरिया में, जहाँ पे जिसमें वो सिंगल सर्कुलर डीएनए जेनेटिक मटेरियल क्रोमोसोम होता है, उसमें क्या होता है, डीएनए एंड हिस्टोन प्रोटीन. डीएनए होता है लेकिन हिस्टोन प्रोटीन नहीं होते. हिस्टोन प्रोटीन्स आर प्रेजेंट इन द यूकैरियोटिक जेनेटिक मटेरियल. They are attached with the genetic DNA of the eukaryotic DNA, or these histone proteins are basic proteins. They possess mainly possessing the basic amino acids arginine and lysine. The histone proteins are H2A, H2B, H3, H4, and they are not present in the nucleoid or genophore. So this is not the right answer. RNA and histone protein, RNA तो न्यूक्लियाइड में होता ही नहीं है, वहाँ पे DNA होता है, तो ना तो RNA होता है ना हिस्टोन. तो आंसर इज राइट हियर. अगेन दिस इज नॉट द. Nucleoid is a single stranded circular DNA molecule. Single stranded circular DNA. Again, the nucleoid is not made up of a single stranded circular DNA. It is made up of a single but double stranded circular molecule. So this is not the right answer. The D option is, it is single circular molecule, single circular chromosome होता है, लेकिन double stranded double helical 2 strands होते हैं DNA में. तो it is the right answer. Next, primary function of bacterial pili is, bacterial pili की main function क्या होती है? Locomotion is carried out by means of the flagella. This is not the right answer. Conjugation, as we know that the these pili are elongated tubular structures made up of a special protein जिनको हम कहते हैं pillin, and there are 1 to 4 pili per cell, and they help in the formation of the conjugation tube through which the genes from mainly the plasmid are transferred from the donor to the recipient. And this process, transferring of the genetic material from the donor to the recipient, is called as the conjugation. This conjugation in bacteria are sometimes called as the sexual reproduction in prokaryotes. So pili are responsible for the conjugation. Motility is mainly by means of the flagella. Attachment with the rock surface is mainly with the help of the fimbriae. So the right answer is conjugation. Next, prokaryotes have 1 envelope system, 2 envelope system, 3 envelope system, D, both B and C. Prokaryotes have only 1 envelope system. They are only covered by means of a plasma membrane. They are not possessing the membrane bound cell organelles as in case of eukaryotes. Eukaryotes have 2 envelope system. One is by means of a cell membrane, जो cell membrane बाहर होता है, अंदर जो दूसरे सब organelles होते हैं उनमें भी membrane होता है. ऐसे such kind of having the 2 coverings, one from the outside, that is the plasma membrane, one for these cell organelles. Such a kind of is called 2 envelope system, that is present in eukaryotics only, while as prokaryotes have only the 1 envelope system. So the right answer is 1 envelope system. Subscribe and share Bioclasses online by Ashik Hussain. Thank you.